was like, okay, look, basketball player. It's just funny because so I have seen them in. They used to little after. They're very good. Much more. That I will need your name, right? Yeah. You understand that? 18, 19, 20. That's 6 years. That's your fucking dog. I feel like I should have one PhD or something. Something big. Yeah. Something. I see you out here, you know. Oh my God. So you know they're you don't have to have like a church to be a CR and you want that. I don't know. Did Heather come back today? Did you get me from that? I thought this was about next week. Yes. Are you going to record this? I love that. It's recording right now. Oh gosh, let me not talk. You know, you got your little. Yeah. I'm going to be at the computer pretty much the whole time you guys get started. Once we get you on your actual lab part of class. Okay, so I'm going to go ahead and get started with the review. We are not going to make it all the way through all the chapters. I haven't made it all the way through any of the chapters, like on any of the reviews. So we'll get through what we get through. I did pretty good on the one this morning. So if we don't do quite as good on this one, you can always go back and watch that one. So I'm hoping we can cover everything we need to here. All right, we're going to start with chapter 6. This is going to be the genetics chapter. You guys know this is the hardest chapter, I would say, easily for most people. We're going to talk about DNA first, and then we're going to get into replication, transcription, translation, operons, and then like mutations and horizontal gene transfer. So there's a lot to cover in chapter 6. It's like the fullest chapter, I feel. Okay, so DNA, we know it's our chromosome, makes up our chromosomes, right? All of our genes are in our DNA. Bacterial chromosomes, they have one single circular chromosome. That's it. Whereas we in our cells as eukaryotes, we have linear chromosomes. We know this because of the X's, right? They're not in a circle. Linear chromosomes, and we have several of them. So it's a big difference there. All right, we have these terms here, phenotype and genotype. When we talk about a phenotype, that's talking about what we're seeing in that person. So if somebody has genes for green and blue, but blue is dominant and will be expressed if it's there, so we would never know that you were carrying around that green gene. But you could pass it on to a kid, and then another person pass on another green to that kid, and they have green. You're carrying it the whole time. So that's your genes versus what you're actually seeing, phenotype, okay? Genes, genotype, what you see, phenotype. Okay. DNA and RNA structure. We have deoxy and we have ribo. That's kind of where these D and R are coming from. So let's start from the top. We know DNA is our genes, it's our chromosomes, we just said. RNA, that's basically our working copy. We take our recipe book of all the recipes ever. We just want to make a souffle. So we find souffle, we make a copy by hand of that out of the cookbook, because remember, it can't leave. So we leave that behind and we take it out and we make our souffle. That's what's happening with all this DNA stuff. It's just basically that. So our working copy that we made, that we're going to take to use to make the souffle with, that's the mRNA, messenger RNA. They're carrying the message to make that gene product, one gene product, okay? All right, tRNA is the transfer RNA. It carries amino acids. So the whole point of mRNA is to carry the message for that one gene. What does that mean? What does it do? What is it for? tRNA comes and matches up to areas. We'll go in more detail here in a couple slides, but matches up in sets of 3. And if it does match up, it pops that specific amino acid at that spot. Then we have amino acid after amino acid after amino acid as we go 3, 3, 3, 3 along that mRNA. That's how that would work. Our RNA is ribosomal RNA. It makes up the ribosome. The ribosome is kind of where this is all going to be going on. It's kind of facilitating this whole process. Okay. That's the idea there. So what are they made of? In DNA, we have a sugar phosphate backbone repeating. The sugar in that sugar phosphate backbone is going to be deoxyribose. Go figure that RNA is going to be ribose for R. Yeah. So the only difference here is we removed like an OH from it. We removed an oxygen, deoxy. Other than that, they're pretty similar. And they have the same kind of backbone. Sugar, phosphate, sugar, phosphate, sugar, phosphate, over and over. But we know DNA is usually going to be double-stranded. And we know RNA is usually single-stranded. All right, in our cells, it's single-stranded. So the idea here is that you know, we keep all of our compendium of our recipes or our blueprints, whatever you like, in that one place and store it there. That is all DNA is for, is to be the book of making things. And if you need to make a thing, you go make a copy of that little gene only. You take it out of the nucleus and then do your business. So you don't need RNA to be double-stranded. It's not holding on to all that information. It only needs to be single-stranded. So only going to be short-lived. All right, then we have the other difference, which is in the bases. We know A, T, C, and G for DNA. Pretty easy one there. Those are our bases that make up what the code is. A binds with T and G binds with C in DNA. RNA is exactly the same, except instead of the T, we have uracil. Uracil is a lot like thymine, the T. It's a lot like that one. It's just a little bit different. It just works better for what RNA does. Okay, so I keep saying, like, they keep making up bases and doing nucleotides, and what is all this stuff? So each unit that is repeating in DNA consists of, we have that sugar phosphate backbone, yeah? A sugar is bound to a nitrogenous base. That thing right there, those three items, makes up a nucleotide. All right, and technically they'll repeat over and over again. The nitrogenous base is what determines whether it is A or T or C or G or U. So that is the nitrogenous base. It's nitrogenous because, go figure, nitrogen. It has nitrogen in it. Nitrogenous. And then we have the phosphate. What kind of charge does phosphate have? Negative charge, yeah? It still always does. It's not going to change. All right, so the other term that we apply to DNA that can sometimes be a little bit confusing for people is going to be anti-parallel. DNA, we said, is two strands, yeah? Complementary across one another. They're anti-parallel due to the fact that the sugars are not symmetrical. If I were to hold up, I mean, I'm always bad about choosing the thing. Maybe this stapler, okay? And could it be any stapler? I know you guys can't see it in this, but I promise there's a stapler here. And you hold it this way. And if this part of the stapler has to have a hand and this part of paper, and you keep having it need to be oriented that way, then you would have to keep going this way, right? So that's due to the orientation of the stapler. However, if the stapler was oriented the other way, the hand would have to go over here, the paper would be going over here. Now we're stapling in the other direction. Literally why it's anti-parallel. Due to the orientation of the sugars. The sugars and how they have their little OHs and all that BS going on, they are oriented in different directions. And they can't be the same direction just because of how they interact with one another. And that's just chemistry. Okay. So anti-parallel means they're running in opposite directions, just like the stapler was. Okay. It means they're opposite directions according to the carbons of the sugar. You don't need to know which carbons or how or why, just because we number it by the carbons. So when we say 5' to 3', we're talking about those carbons and those sugars that can only be oriented kind of one way on each strand. All right, so everything will be moving as we go through this from 5', which is usually what we consider to be the beginning of the strand, to 3', which is towards the end of the strand. Okay, if we have nucleotide after nucleotide, there will be a 5' up here. Add on some nucleotides. Down the line at the very end of that nucleotide set is the 3'. So it can need to add another nucleotide onto that set, you would be adding it to the 3'. You see what I mean? 3' is always at the end. And so it's just like you could think about if you're working on this with like Twizzlers or something like that as you bite along it. But so yeah, anti-parallel, opposite directions. When we go through anything doing with the DNA, replication, transcription, whatever it is, always moving 5' to 3'. It's just the orientation of the backbone. You don't even know why or how. That's what's happening. And that's why it works that way. And how we control it, it's important for that. So we just said that part. We said anti-parallel, dealing with that backbone being opposite directions. That's important to know because as we are making copies of our DNA, we can only add on nucleotides in that 5' to 3' direction. So onto the back of a 3', we can add on another one and then keep adding and adding and adding so that the 5' on the left and the 3' on the right, and we just keep adding another one onto that 3' end and keep building. However, we are anti-parallel, and you'll see in the picture, it'll make more sense. But let me just kind of get it out. Because we're going opposite directions, as you are opening up those two strands that we have with the DNA, one can go straight in 5' to 3'. The other one, you can't go that way on the other one. It's oriented the opposite way. So what do? You can only go 5' to 3' direction, right? So what do? How do you build from that strand? You build it in pieces. So as we open up, we opened up a little bit. Now there's more space here. We can start building from there, make a piece. It opens up further, little space open, start from there, make a piece. And eventually just link all those pieces together. Those pieces are called Okazaki fragments. It's just the name of them. A guy probably, discovered it or something. The one that has to be made into pieces is called the lagging strand because it's lagging behind the leading strand, which is just going straight on through. And when, of course, we have pieces, we have to seal them back together. Anytime you seal something together, that's called ligation. To ligate something is to join something together. So that's why we call it DNA ligase. We're joining the pieces together. They are called phosphodiester bonds that we're forming there, but you don't need to really worry about that right now. Let's watch this video. The video is helpful, I promise. It's long, it's like 6 minutes, but bear with me. It's worth it. This is the only speakers I have, I apologize. Cool. But that helps. I feel like seeing it. It kind of helps see everything and how it works. There, I have another video. It is basically the same thing, but like more linear, and it also has closed caption on it. So, okay, I said it did. It did the other time I played it. Whatever. I can't do anything about that. It's kind of nuts when you look at that and you see like all those things that work together on that lagging strand. Anyways, that's that. So moving on to transcription. Making RNA from DNA is called transcription. Like I said, you've got your recipe book. You want to go make a copy of a recipe. Then you have to write it down by hand. And when you write things, that's like scribe. Yeah, we're transcribing it. Anytime any kind of RNA is made, we have to transcribe it from DNA. So DNA will let us make mRNA, tRNA, or rRNA. All right. That mRNA is our working copy. Like I said, we can carry it out of the nucleus to actually make our product, whatever that is. It has 3 steps to it: initiation, elongation, termination. And there's no weird other thing going on another strand or anything like that. It's just that simple. Start, go through it, and then be done. I like it because it's easy. So in our cells, this is going to happen in the nucleus because we have the DNA there. So if you're going to operate off of the DNA to make your copy, you have to be doing that in the nucleus. Bacteria don't have a nucleus, so they get to kind of do that out in the cytoplasm or in the area where the DNA hangs out, called the nucleoid region. We already talked about A to T and G to C, or replace the T with a U, but it's the same concept. That whole interaction is how we're going to start adding the nucleotides for the mRNA. Okay, that's going to make us our working copy of the gene. Let's watch our video for it. I like this one, it's easy.
During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in 3 stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other.
I think that's pretty straightforward. So as far as that goes, adding the nucleotides that are complementary, and then making our usable messenger RNA that's going to carry that message for your product. All right, so let's go on to translation. Now we've got that messenger RNA carrying that message, that written down copy of that exact recipe that you copied out of the recipe book. And now we want to make our souffle or whatever. Okay. In mRNA, we have these 3 nucleotide sets, always in 3s. In DNA, the DNA version is called a triplet. In our mRNA, the messenger, we call it a codon. Each codon codes for a specific amino acid. We can even have charts of them. If you have AAA, then it will code for this amino acid. And different from GGG, it codes for that amino acid. There's 20 different amino acids, and there is some overlap to where sometimes like AAA and AAG might code for the same amino acid. You don't need to worry about which ones do or don't, but that helps if we have any sort of mutations, help kind of smooth that over a little bit. It's a nice little, they call it the wobble effect. But yeah, so a little bit of give there. All right, on tRNA, we have the anticodon, and yeah, it specifically is the complement to the codon from the mRNA. The 3 nucleotides like AAA on that mRNA, A would match up with the UUU on that tRNA. Exactly the opposite of it. So they match up, and if they do match up, that's going to say to those molecules, this is the correct amino acid that I'm carrying. We'll add this to your whole lineup, and it'll keep going on. So they carry that amino acid for that specific code, and then we'll be linking them together by a peptide bond to create a whole protein. All right, this does play. I made notes for myself just because I thought they wouldn't play, but they ended up being able to. This is a pretty good one as well. Once it gets going, it's a pretty good one. You don't need to know the names of the sites. I just want you to see the process of this. So basically it's going to match up at that first space, shift over. That's where it's going to associate with the other amino acid next to it. It becomes blank, knocks out at that last spot, bring in the next one as we shift down and keep on moving through. Just keep on linking those amino acids. This is where that stop codon comes in. So the stop codon basically doesn't code for an amino acid at all. It just makes the ribosome dissociate, like it comes apart because of it. So that's important to remember when we come back to it when we get to the mutations. First, oh, the fun we'll have with operons. Operons, only bacteria have operons. We do not have operons. It's not really too crazy of an idea. It's just a set of genes. You know how I say an mRNA codes for one gene? We copied one gene out. In bacteria, you can have a whole bunch linked together. They just have them all come out at the same time, that's all. So any of them that are linked to doing the same thing can be controlled by the trigger. And basically that's how bacteria would, control what's going on in their cells. So we'll start with, responding to our environment. We'll start with our repressible operons. Now, repress, like as though you were going to like repress your feelings or something like that, needs to kind of halt or stop. If we can stop it, it had to be going. Okay, so it's repressible. Basically, you can see it as always on. We can turn it off. All right, our factory. We have a factory that makes pants and is churning out these pants like crazy, and people are buying them like crazy. And it's wintertime and it's cold, and everybody wants to wear pants. Making pants, making pants, making pants. Starts getting warm outside. People want to wear shorts now. And now we have too many pants sitting around, right, at that factory. So the idea is that the pants can be brought to the repressor or the manager, maybe, however you want to look at it. And once that happens, it's going to put halt to making things. Okay? Give the manager the pants. He comes down and tells everybody stop. That is essentially what's happening here. If we take the molecule product, bind it with that repressor, it's just a protein. Anytime a protein binds with another thing, it always changes its little shape. So we got our shape change going on. Then we can bind onto the site on the DNA, and no longer can we make anything. We've lost it. So the manager comes down, shuts down the whole factory. That's repressible. Inducible, kind of the opposite side of that coin. Okay. The famous one associated here, the lac operon, lac for lactose. Most bacteria would prefer to use glucose as their energy source. That's not surprising, considering that if you learned anything about metabolism in the metabolism chapter, glucose is like the #1 for making energy. So now, though, this bacteria is going to have to use lactose. So if you are inducible, if you can be induced, you weren't doing the thing, right? We're going to induce you to do the thing. Think of it even as going to have a baby. The reason they induce you is because you're not doing it. So they come and they push you along. So that's inducible. We have to turn it on. We're only going to make genes to break down the lactose if the lactose is present. That just saves the bacteria energy. So what happens here is that repressor protein, it's like the manager is sitting there waiting. And when he gets a call from the stores, giving him lactose to say, let's go, giving him money, say that's that. When he gets that money in place, which is the lactose here, it associates with him, and now he'll go and tell everybody, start up the factory again. Okay, it's like that. Repressor binds with lactose. When it binds, it changes the shape. They will do. It comes off of the DNA, and now we can start making the stuff. Okay, that's repressible versus inducible operon. The place where the repressor that's making this, controlling all of this, binds onto the DNA it's called the operator. Okay, it doesn't do really anything else in any other setting other than an operon. So I feel like those words go together pretty well. I'm not gonna make you watch this. It's like the worst thing you'll ever have to sit through. It has way too much energy, information. I watched it and I was like, what? You don't need to know it. All right, the last little bit in the genetics chapter. Mutations. We can have a point mutation, exactly what it sounds like. One little A gets changed to something else. It can have a pretty profound impact on things, but that's a point mutation. One nucleotide change. All right, there's two kinds of things that people easily confuse with one another. Missense and nonsense. Missense, think of it as like being a misfire. You didn't quite hit the mark, let's say that. It's a different amino acid than what you were supposed to be coding for. So you had a change in a nucleotide, maybe A to G, it doesn't matter. But now we're not coding for that same amino acid. We're doing a different one. We learned in our chemistry stuff, if you have like a negative charge, but now we had a change here. Now we have a positive charge amino acid, massive change in the structure of the protein because of how it might interact. Wrong amino acid from the missense. Worse than that is the nonsense. We have no sense going on here at all. We're not going to get any amino acid out of this. And the things that we don't get amino acid out of, those are stop codons, right? So there's some sort of change went on that we changed from coding for an amino acid normally and keep going with that. Now we have a mutation that we've changed into a stop codon. You're stopping the production of your protein. It's like stopping making your genes, like with just the freaking cat pockets or something. Useless. So that's the idea. Pretty dangerous stuff if you have that coming out. Next, we have silent. Like I said, some of the, like AAA or AAU can code for the same amino acid. So if you have a change that has the same amino acid, that's silent. You would not know based on the amino acid sequence. Next, we have a back mutation. Let's say it's like we're looking at a population over time. Usually bacteria, they grow pretty quick, right? They've got a lot, they can grow real fast. So you look at the original bacteria, it was blue bacteria. There was a weird mutation that made it turn green. Now maybe those green guys had the mutation go back to blue. That's a back mutation. Went back to the original. All right, frame shift mutations. Anytime we have like an addition or a deletion of like a single nucleotide, that's what we're talking about here. So if we read this in sets of 3, AAA, GGG, CCC, and I tell you that AAA codes for blue and GGG codes for green and CCC codes for red. Well, if you delete that third A, it's still going to shift everything over as far as how it reads it. So we're still reading in 3s. We don't have AAA anymore. We have AAG now. If we deleted an A, where that line is. So that's not going to make the same amino acid probably. And if you keep going on down the line, look at that, like GGC, and then the CC will have something else going on after it. I don't know. This can cause massive, massive, massive destruction just from removing or adding in one nucleotide to the protein. Imagine this, if you were reading a book, you're a couple chapters in, and some magical wizard comes along and takes out one letter from one of your words, just one letter, shifts all the other letters from the entire book up, but keeps the same number of letters in each, like what would have been a word from before. That's like what's going on here. You could not read that book after that. It would be done. Like it would just be a hot mess. So that's the idea here, especially if you rely on reading something in order. Frame shift, horrible, horrible thing. You can see how that and nonsense could be like the worst ones you could have. All right. Bacterial horizontal gene transfer. We talked about this in the transformation experiment in the lab. Like just like, what was it, Tuesday? So here we've got transformation, where we pick up that little DNA from outside of us, exogenous, right? Competent cells are able to pick up exogenous DNA, and that is called transformation. Transduction, this is whenever we have a phage, which is a virus that infects bacteria, and that phage DNA sticks itself into the bacterial DNA and becomes like one with it. It's called a prophage at that point. And if it has extra genes in it that it's carrying along, those genes can make the bacteria like more, virulent or like more toxic or make a new toxin or whatever, be able to survive different conditions, blah, blah, blah. It can make the bacteria a lot more pathogenic, a lot more virulent. When that happens, we get those special traits in our bacteria because the virus put that DNA in there. That's called lysogenic conversion. We talked about that in the first one, in the virology chapter, but that's kind of where this is going to tie back in, okay? The last one is called conjugation. Just like a conjugal visit, you can think of sex with this one. That's exactly what's going on. Bacteria sex. They're going to form the sex pilus. It's called a pilus. It's like a tunnel between two cells so they can transfer their DNA. That's the way that bacteria can transfer DNA between each other. All right, that's chapter 6. Anything that you guys have to ask about the actual like genetics straight up? That stuff.
Still kind of lost on that one.
The operons? Okay, so let's go back to it so I have the slide up so I can talk to you guys about this. Okay, so operons, like when you see on the board, now this is going to suck for people that are, I can't draw on that very well. I don't have my little pad. But imagine you guys are watching this, it's still amazing. If it was on like our little DNA circle here, that's what bacteria have. Circle. And you can imagine here, there's like a zone here. It's called the operator. And it's blocking where the promoter further up would be. So what wants to happen is bind onto promoter. That's where RNA polymerase would bind. So if we're making mRNA, this is DNA, right? If we're making our mRNA copy, a little recipe to take with us, we need RNA polymerase. It binds to a promoter. Next to it, we have this operator. And downstream from that, we have these genes that do whatever for whatever you're doing. Okay. All right. So it could be a set. In bacteria, they can have more than one gene going on there. You can't do that, but bacteria can. Okay. So the repressor is like a guy. Do it purple so you know he's going to associate with the operator. Wish this marker worked better. So the repressor. In the case of a repressible operon, this is always working. Okay? This can go through. It's not being blocked by anything right here. So we can have our little guy going through and make mRNA, make mRNA, make mRNA, constantly churning out the written down recipe so we can make our product. All right. So we can turn it off though. Our product, like I said, pants or something, right? Let's say this codes for some pants. We're building up the pants over here. Genes for pants. Now we have a repressor protein. The pants come along. Here's some pants. And that product can associate with that repressor. In this case, now when it's with the pants, it can now bind at the operator. Here. Pants plus repressor, we can now stick down at that operator. This was us making a factory of making pants. This is the manager just hanging out, probably doing nothing because that's what managers do. And somebody brought him some pants, and we got too many of these. Now he's going to bring it down to the floor and stop operation here. So now our repressor, because it bound with the pants, changes its little shape, and now it can bind here at the operator. And literally, we cannot move to make any DNA here. So that is repressible. So it's on the whole time. We're waiting for too much excess product. When we have it binds with the repressor, there's enough to bind with that. We can go and sit on that operator and block making that product. We don't need to make any more. We've got too much of it. Okay, that's repressible. All right, let's do a different purple. This one's worst. Here is the same thing. Do a couple layers so you know that it's DNA, double stranded. Here is our operator. Here's our operator, same thing. Here's our promoter region where the RNA polymerase will bind to make mRNA, drive this whole process. And then here are all the genes associated with it afterwards. In this case, let's say there are genes to break down. Do you guys remember the word for that in metabolism? That would be called catabolism, right? Break down lactose. We don't see this sugar a lot as a bacteria, so we don't always have that running. It's not going to be like the other ones, constantly running. Only going to turn it on whenever it's there. So we got lactose coming along. We had that was like there's a whole separate thing here now. Of course, I had to do that. have lactose. This one, because we don't need it all the time, we don't need to be making it, wasting our resources. So our repressor, our manager, is telling us we don't make this unless we are paid to make it. Okay? This is they're waiting, blocking this, so we do not make it until we are specifically told to. In this case, our lactose comes along. Lactose sugar comes here over to this guy. Pretend that he's being given money or something, whatever. He's really interested in this. Lactose comes and associates with this repressor. He changes the shape, gets real interested, walks away, and then now calls to turn on the factory. He got paid. Okay. The lactose is there and it turns this whole thing on and it works in the favor of the bacteria because it's going to break down lactose now. It's being controlled by it. So if we break down all of it, what's going to happen? The repressor's going to go back, right? When it's done. We've broken down everything. We'll shut it back off and wait till we get more money, more lactose. Okay? So that is what we would call an inducible operon. It's not on all the time. You have to turn it on. Yeah. I think that's okay. All right, do you have a question about it? I don't know if that helped at all, but like, I don't know any other way to explain it. There's a specific part that is confusing. I'll try to walk through that part. I don't know. All I can think of is remember everything that your cells do is driven by protein. Always. 100% of everything your cells are going to do is driven by protein. So if you're going to do anything with your cell, break down lactose or stop breaking down lactose, it's going to be driven by an enzyme somewhere, a protein. In this case, our protein is being regulated by lactose, right? Or any other product that you can think of. And then based on that, how that operon is set up, or what that cell needs, it may be repressible or it may be inducible. I want to be clear. These are not the same operons, okay? They are completely different gene areas. So it's not like a inducible operon can sometimes also be a repressible. That's not how it works. For one or the other. Okay. That's all I got. Okay, moving on to gel electrophoresis. So DNA has that negative charge from that phosphate, right? We know sugar, phosphate, sugar, phosphate, sugar, phosphate, all along its little backbone. So that phosphate gives it that negative charge that's going to make it move to a positive. Like always, how everything works in chemistry and physics. Now we remember that pieces of DNA, we call that measured in a size called base pairs. And that's literally referring to the bases of the DNA. That's how we determine the size. How many bases are there? Base pair. So BP means base pair. So like 50 BP means 50 base pairs, literally. If you see 50 kBP, that's 50,000 base pairs, okay? Just like a kilometer, K is 1,000. All right, smaller base pairs will move faster through that little gauntlet of the gel that we've set up. And that's what the whole thing is moving through. So you can imagine if there's a whole bunch of linebackers, the smaller guys are going to get through easier than the other big guys. Yeah. So that's that idea. The larger ones will get stuck. They'll stay up at the top. The smaller ones will move through and go more towards the bottom. And the variations in the middle. Yeah. So that's gel electrophoresis. You need to know that concept for both the lecture and the lab. PCR, polymerase chain reaction, another concept you want to know for the lecture and the lab. This is just DNA, making copies of DNA. So each cycle that we run of the steps down there is going to double what we had originally. So the steps are denature using high heat to split those double strands of DNA apart DNA strands are held together by hydrogen bonds. They're weak bonds, so it's easy to break them apart with heat. So those strands will split apart. Then we can stick on primers. We saw them talking about that before, right? DNA polymerase, when we're making new DNA, has to build off of something. It just doesn't build off of the friggin' ether. So it has something to build off of. So we'll add a little primers that we designed and happen to match up very specifically with certain genes. We can target certain things. Then we're going to add on nucleotide with DNA polymerase and make a copy of that. Okay, so we go from one double-stranded molecule that will come apart to make two single strands from the heat. We'll have little primers to start us out, and then we'll go along and make little copies along the way. And now we have two double-stranded pieces. That's really all you need to know about how PCR is going to work as far as determining, if I ask you on any of these tests, how many pieces of DNA would you have if you start with one and you go through 3 cycles? You don't have to do the math. I can't do the math either. Just draw them out, okay? Draw out the little lines that represent it and then count how many. If every time you go, 1 goes to 2, anytime that you see a 1, it'll go to 2. So you'll just have to think of it that way. Or you think of it as just doubling at every time. So 1 becomes 2, 2 will then become 2 and 2, which is 4, right? And then you have 2, 2, 2, 2. That would be 8. And you can think of it that way, or you can just draw it out, whatever works for you. But yes, you will have to be able to know that. I'm not going to give you like 32 cycles. Okay, it can be like 3 or 4, something like that. Okay, cloning. This is just talking about taking DNA that we're interested in and that we have understood, now putting it into another form so we can use it differently. So playing with DNA and other DNA, basically. Vectors are the means by which we do this. We accomplish this. When we did our transformation experiment, we had a plasmid, that little circle of DNA that had extra genes on it. And we can give it like antibiotic resistant, like we did with our transformation experiment. And we can give it like human insulin. Think of it as when we did our experiment, then we talked about GFP, that green fluorescent protein was our product. It could have just as easily been human insulin. Okay, it could have been. So that's actually how they make insulin, if you need it. So yeah, so that's the idea of how that works. That's the plasmid type of it. We also have viruses. We can take human infectious viruses, clear out their inside genomes, and put the genes we want so that they can do work for us. Go infect cells and put new good DNA in there, like we were saying with the gene therapy stuff. So we often use viruses for that. Don't forget about that. We forgot. I mean, I feel like it's been like so long ago since we've talked about gene therapy. Artificial chromosomes are exactly what they sound like, just making it from scratch. We don't talk about that much. All right, next we have transgenic. That's the same thing as a GMO, genetically modified organism. Anytime that you have taken an organism and put different genes in there, then that's transgenic or genetically modified. Then we also talked about restriction endonucleases in this chapter. It's kind of, we talked about at the beginning, but in the study guide, it was at the end. I wanted to keep this in line with the study guide. So restriction endonucleases, they were enzymes, ACE, right? Anything with ACE at the end, enzyme. They were enzymes that helped get rid of foreign DNA normally in bacteria. That's what they would do. But now we figured out they bind at these weird sequences that are palindromic, the same forward as backwards. That's not common in DNA in general. So we can target specific sites because of that with these guys. And each one is like a response to a different kind of sequence and all of this. But whatever it is, they can make cuts and leave a single-stranded part at each end. You can see how that single-stranded part wants to bind the A to the T and the G to the C and all of that, sticking out there waiting. So you can cut your little plasmid with your enzyme and introduce your new human insulin gene that we also cut with that same enzyme. We made it that way. And I can stick it in there on those little sticky ends that we've made. And we can seal it up with our good old friend DNA ligase. and pop it into bacteria through transformation like we did in our lab. That's how that would have been made. So that's the idea. They create those sticky ends, which is single strand bit that overhangs. Okay. Because we're not going to be able to get through all of these chapters. I've been asking the other classes kind of what they want to go over. So we did 6 and 8, which I feel like are kind of the same. Much more of 6, just leaving on. We can go chapter 9, which is nutrition. Chapter 25 is like the environmental and water treatment stuff. 25, yeah, that was 25. 10 is metabolism. That's a big one too. Then we have 13 and 14. I personally feel like 13 and 14 are just vocab tests. Okay. So I feel like if you want me to go over them, I will, but you better have like specific concepts in mind. I don't want to just read you vocabulary. So do you guys want me to go to 10 first? Okay. That's a popular choice. Okay. And then I would like, if we have the time after that, I want to go to 25 so I can show you what you should not be studying. That's more or less what chapter 25 is for. All right, metabolism. That's just a term talking about the chemical reactions in the body, just all of them all at once. We have two main types of reactions. You got to have building things and breaking things down. Building things up, we've got anabolism. We need energy to do that, right? We want to tear things apart. We can steal energy from that process, which is really literally what the whole aerobic respiration is about. But before we get to that, catabolism really is referring to any time that we're going to be breaking down molecules, so bonds in general. All right, so I remember it again. Cat. This cat is able to destroy. Cat-able catabolism. So I remember that means destroying things. I don't know what anabol does, but it's not destroying things. Enzymes. They're proteins, first of all. They're a type of protein that's going to help chemical reactions go. Overcoming activation energy is what they call it in chemistry. So it's the amount of energy required to make the thing happen. Enzymes help that, so we need less. That's their whole purpose. That's like pretty much what most of your genome is going to code for. Now we have a holoenzyme, which is exactly what it sounds like. Holoenzyme. It's going to be the whole enzyme. Okay, the whole working bit of that enzyme, holoenzyme. It consists of the protein part, which is we call it the apoenzyme. I don't have a good memory one for that one, sorry. And then we have the cofactor. The cofactor is just the part that it needed to work. So that could be a little molecule as small as like a metallic ion, like iron or something. But it also could be these other organic molecules, like vitamins and stuff like that, that this enzyme can't work without that. So then it's called a coenzyme if it's an organic molecule. The inorganic, we just call it a cofactor. A coenzyme is a type of cofactor that is organic molecule. Okay, here's the enzyme types. The only one that I really want you to be like super worried about what it really means at its little tiny core is the oxidoreductase. So you need to probably be aware that a ligase will create bonds. I mean, if you don't know that by now, I keep talking about DNA ligase. But and we'll talk about hydrolysis in the lab, and it's just breaking down like starch, like hydrolyzing starch or whatever. Are we building starch in those experiments? Absolutely not, right? We're gonna break it down. We're gonna do. So hydrolysis is breaking things down. We're gonna use water up while we do that. A ligase creating bonds. We'll also create water as a byproduct. So they're kind of like opposites of each other. Okay. I'll get back to oxidoreductase, I promise. Transferases move groups around from one place to another. Isomerase changes the shape. Liases, we got some double bond action going on. What I said to you about these things is all you need to know about these things. You don't need to know anymore. Oxidoreductase, that enzyme, they're enzymes. Anytime you have a redox reaction, if you've taken chemistry, you've probably heard of redox. We're talking about reduction and oxidation. You can't have one without the other. They always come in a pair. So oxidoreductase, they always are going to come together. We're talking about moving electrons around, shifting electrons. And if you remember what we're getting ready to talk about with making our energy, that's exactly what we're getting ready to do, is shift electrons around. So that's important for this. So anytime that there is an electron exchange going on, oxidoreductases are going to be involved. That's the enzyme involved in that. Okay, the concept of a redox reaction is important as well. You need to understand what is reduced and what is oxidized. And the way I remember it is Leo says Gur. And if you can just remember, if you know if Leo says something, at least you got the Leo down, because you could say Leo, okay, what was that? That was loss of electrons means oxidized. So if we lose electrons and that means oxidized, that must mean that reducing is gaining electrons, which it is. Gain electrons will be reduced. So Gur, that's the Gur part. Leo says Gur, or you can do oil rig, it's the same concept, okay? But yes, if we gain an electron, I would say that molecule is reduced. Okay, regulation of all this stuff, we already just talked about trying to regulate things at the genetic level. That's for bacteria over there. For our cells, we can regulate it the similar way, but we just don't have a whole bunch of genes in a row. When we are regulating something at the protein level, though, like the actual enzyme and how we keep it from doing or make it do the thing, here we're talking about competitive versus non-competitive inhibition. Enzymes act on a thing that we call a substrate, right? That's the whole thing that it will interact with that's important. So if the thing that's blocking, which we call the inhibitor, binds where the substrate should be, so the substrate can't get in, like standing in the doorway, blocking, that inhibitor, that situation we call competitive inhibition. So you're trying to get in to that same spot, and they took it up, and now you can't do it. All right, non-competitive, it's sort of like what we were talking about with the genes and everything like that over the pants and regulating that. But basically non-competitive inhibition, we're enzyme, we're making a product with our substrate, cranking along. We usually will have a product that we make too much of, like too much of the pants, and it'll come and interact. So if I were that enzyme, it comes and binds with me, my product of what I'm already making. It's like it's tapping me on the shoulder and saying, "Uh-uh, we don't need more. I'm already here. I'm not getting used up. You need to stop making things." The same thing. So we're not binding at the active site. We're binding somewhere else on that protein, causing it to change shape. Like if you're tapping on the shoulder and you turned, now you can't do whatever the thing was maybe that you were doing. It's just that simple. Competitive versus non-competitive. Whether you are competing with the actual site where the reaction's happening. Respiration, this is creating energy. ATP is our energy molecule. It stands for adenosine triphosphate. When you talk about ATCG in DNA, A is adenine. Okay, so adenosine is adenine with a sugar. So this looks an awful lot like a nucleotide, right? Adenine, nitrogenous base, sugar, ribose. And then we have a phosphate. We have 3 of them here. But if we had just one, that would just be a nucleotide. So it's exactly like a nucleotide, so it is related to DNA. In this case, we have 3 phosphates. They're super negatively charged, so they're going to repel one another. We're building a lot of tension. We can break that bond, and that can cause us to release energy we can harness. What we're about to do is make more of it, so our body can continue doing the things. So that's ATP and what it's about. We have different ways that we can make ATP as an organism: aerobic, anaerobic, and fermentation. Aerobic, obviously, we're going to have oxygen involved. Aero, that's going to make the most energy by far. Okay, I say by far, because anaerobic, we're not using oxygen. We can make quite a bit of energy. Sometimes it doesn't make very much, though. It just depends on the bacteria. Next, we have fermentation. We only do the first set of steps in fermentation. It's called glycolysis, and we just repeat it over and over again. You don't get very much ATP, but you can live without oxygen like that. And then we have NAD and FAD. These are our electron carriers. Now, you see NAD plus and FAD plus. we're going to stack onto these guys some electrons in the form of a hydrogen that has an electron with it. So that's how we look at things when we're doing these reactions. That hydrogen with the electron is not H plus. Got an electron. It's just an H. So NADH and FADH2, when they're reduced, right, they gained electrons. And H comes with electrons. So we also have H's that do not have electrons. We call those protons. We know that those can happen in the cell too. But when they have electrons. All right, so we've got 3 major sets of steps in aerobic respiration. Glycolysis, Krebs, electron transport chain. Guess what's going to get transported in that? Electron, right? So that's the whole goal. That's the bottom. That's the big daddy. That's our goal. So you can already say, okay, the last thing was called the electron transport chain, electron, even if you've got electron something. Okay, that's the last one. So if we're moving electrons, we have to get electrons to do that. That's the whole point of everything above. The whole point. Okay. Glycolysis. The point of glycolysis is to make pyruvate. That's the only thing it's doing that we care about. Does it make ATP? Yeah, a little bit along the way. Does it make NADH? That's our electrons, right? Yeah, it makes some. But we really want that pyruvate because that pyruvate is going to go eventually down to where the big daddy is. Okay. Who cares over that ATP? Made a couple ATP. So we're taking glycolysis, where we have glucose. That's what we start with typically. That's the textbook. And it's 6 carbons on there, breaking it apart into 2 3-carbon pyruvate molecules. Pyruvate and pyruvic acid are the exact same thing. So we've got 2 3-carbons from the 6 carbon. Pretty easy concept going with that. All right, Krebs cycle. We're going to take that pyruvate, turn it into acetyl-CoA. Then you don't need to remember these and fill in blanks, right? You need to recognize them as well. Pyruvate is 3 carbons. Acetyl-CoA is 2. So the other carbon, where did it go? We're kicking it off as carbon dioxide. When you kick off some carbon like that, you release some electrons. And guess what's up there to pick them up? NAD. So we got NADH. Now, only a little bit of that going on. That's just a little intermediate step. But it's still important. We've got a stupid little 2-carbon acetyl-CoA. There's just these really wonderful, confusing organic molecules that are in the Krebs cycle that basically acetyl-CoA is going to climb all around. And we're going to kick off some carbons while we do it. So here is what's going on in a Krebs cycle. You put that 2-carbon acetyl-CoA, it dances with some other organic molecules. We're kicking off some carbon dioxide while we're kicking off our carbons. We're also going to take with us electrons. We're also going to make a little bit of ATP along the way because why not, right? So that's the idea. The main goal of Krebs, electron carriers, NADH and NADH2. If they are NADH and FADH2, then they have been reduced. They're carrying electrons. They've gained electrons. That's the whole point of it. Glycolysis gave us pyruvate. We turned it into a molecule that let Krebs turn that into electron carriers. That's what happened here. So now we got our electron carriers because, like we said, we're fixing to take a ride down that electron transport chain. So what's going to happen here? We're going to take those negatively charged electrons and pass them through these other molecules called cytochromes. You don't need to know too much detail about that, but that's where the electrons are going across the membrane. This all happening on the membrane. So transfer those negative electrons through those. They're getting excited and everything. Ooh, we're getting touched by this electron, negatively charged. We like the negative guys. Then we're going to pump out protons, which are H plus, because remember, now we have our electron going across this way. What did we leave behind? The hydrogen. It's not there, or it's not attached anymore. We're straight up electrons now. So now it's an H plus. What we got? So it's positively charged, right? So of course there's going to be interaction going on here. As the negative goes across all of this way, it's going to force the positives to get transferred out of the cell or across the membrane. Across that membrane, positive, 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 as the negative, negative, negative across. Positive out, negative across. That's called chemiosmosis, forcing those positives out because of the negatives going across, chemiosmosis. Okay, so we're moving our electrons down. We're pumping our positive protons out, and they're building up and they're building up. That's what the proton motive force is, when we have all those positives outside. Now, we have a bunch of positive outside. That makes us probably negative on the inside. They want to come back in, right? Just like everything else in nature, they want to be equal on both sides. In physics, we call that entropy. So it's just like diffusion. Same idea. So it wants to spread out, be equal. So they want to come back in. They can't just cross across the membrane without some help. We got over the one side with help, with the electrons. If we want to come back, we need more help. So what we do is we go through this special tunnel, and when we go through that tunnel, it's a protein. When we go through that protein, because we're little positive guys, and we know how positive can affect things, yeah? Charged things can have quite an effect on other molecules. These positively charged guys cause that protein to do a thing. And what that protein does is put a phosphate group onto ADP and turn it into ATP. So that is called ATP synthase. That's what it sounds like as the protons come in, forcing us to start producing ATP. Okay, so that is basically how the ATP is being made in the electron transport chain. We will make 32 to 34 ATP in this one sitting from one glucose molecule. That's nuts, because I just told you up here with the glycolysis, we made a little, Krebs made a little by a little. I mean, 2 net in glycolysis, 2. In Krebs cycle, 1 per pyruvate, so 2 per glucose, right? 32 to 34 in electron transport. You see now why that's a big deal to make the pyruvate and everything. We're not just, futzing around. Goal there. It was worth it. So that whole process, because it involves moving around electrons, oxidative phosphorylation is what it's called. Yeah? Oxidative electrons were involved. So much you know about it. Thanks. Okay, that's how that was made. We got a problem though. So we made this stuff. Now we have to maintain house. We've got all these electrons that we've been shuttling down here. If we just shuttle down electrons, they don't go away, they don't disappear to the ether, right? So they have to go somewhere, or else they're going to build up. Same thing with the proton. You're not going to be able to keep, bringing them back in if you don't have that disparity of charges, right? Positive and negative. If it's positive and positive, you ain't moving anything across. So in order to maintain house here, we have to get rid of those positive protons, the electrons themselves. And what we do is, remember how we had the H's that were just H's because they had electrons? We're bringing them back together. Make H plus, take an electron, now we have just regular H. Now we have 2 of them. Now we take half of an oxygen molecule. Now we have water. We took care of it. It's done. That's why we breathe in oxygen. That's the only reason why we breathe in oxygen, right there. So that's where the oxygen goes whenever we breathe in. So yeah, it's important. So if we don't get oxygen, we can't make sure that the protons are getting taken care of, or the electrons are getting taken care of, and therefore we are not going to be able to make energy, and it does not take very long for you to die from that. That's how important all of this is. Okay. In eukaryotic cells, in our cells, we've already talked about how mitochondria are the powerhouse. So that's where all this is happening. In the folds inside there, we call them the cristae. And the inside we call the matrix. It's not like the matrix in the movies. So the cristae are the folds of membranes. So we just have this going on all along those membranes, and that's what those are for. In bacteria, they don't have organelles. They don't have that. So basically, they're doing all of this on their inner membrane that we call the cytoplasmic membrane. So when it touches the cytoplasm side. So yes, they kind of have to sacrifice inner membrane space for that, which does kind of suck for them because pretty much everything goes on along that membrane for them because they are bacteria. But yeah, so eukaryotes in the mitochondria on the membrane called the cristae. And then bacteria is going to be in the membrane, inner membrane. All right, then there's our little complementary system that's going on. Photosynthesis. We have a light reaction and a quote unquote dark reaction. The light reactions have to have light. The dark reactions can occur just fine in light. They just don't need the light to go, right? The light reactions cannot happen in the dark. So the light reactions, we take photons. Those are going to excite pigments, which we know is like chlorophyll. Those get excited the same way the cytochrome guys did in our respiration. So they're going to start pumping out freaking hydrogen ions the same way that they did in respiration. Just now we're using photons instead of electrons, basically, and pigments instead of cytochromes. Anyways, so we create that gradient. They want to come back in. They go through ATP synthase. We make a little bit of ATP. It doesn't make very much, but it makes enough that we can then take that ATP to our dark reactions. In there, we are going to fix carbon dioxide, which is inorganic, and turn it into glucose using the ATP that we made. That's the goal. Why is that the goal? Well, when we go up here and make our little bit of ATP, it's like 2 ATP, I think. Maybe it's 4. I can't remember. It doesn't matter. I don't need you to be tested on it. Little bit of ATP, 2 to 4 ATP. That's not much, right? Wouldn't they much rather have the 32 to 34? Yeah, of course they would. That's what they're going to do. They made glucose right here just to do that. So now they can shuttle that back through cellular respiration, just like we do, to make all of their energy. Yep, plants have mitochondria in addition to chloroplasts. Okay, that is chapter 10 and chapter 6 and 8. Chapter 13, vocab stuff. Chapter 14, definitely vocab stuff. We could go back. I want to go back to 25 really quick. I'm just going to kind of really drive this home. All right, because I did all of this based on the study guide. Here is what on the study guide I marked out, because maybe people are going through this and they're like, talk about this. What the heck is this? So I'll tell you this. Number 1, no, we're not doing it. Number 2, no, we're not doing it.
Are you on the chapter 25?
25, yeah. 1, 2, 4, 7, 11. We're not doing them on 25. We didn't talk about them. Don't worry about it. So start with number 3, that metagenomic analysis. We only just kind of talked about that. It's just basically like letting you get sequences of stuff so you can start comparing things and make assumptions about it. That's all that is talking about. That's not really meant to be getting into crazy stuff. The only next part that I really care about is number 5, the biogeochemical cycles. We talked about carbon and nitrogen. That's the ones I want you to worry about. Carbon, we just talked about a little bit with that photosynthesis. Yeah. Carbon dioxide, plants take it, turn it into organic molecule. We eat the plant, we exhale carbon dioxide, plant takes it in, makes the organic molecule, we eat it, and so on and so forth. That's the carbon cycle. I don't need you to be more complicated than that. Nitrogen, the nitrogen cycle. It's important for the plants. Bacteria provide nitrogen to plants. They turn it into a form that plants can use. Without the bacteria, the plants cannot grow. So what do the bacteria do? They do these processes. Now, there's ammonia involved, there's nitrate involved, there's nitrite involved, and all of that. I don't need you to know which is which. The terms, I'd be like recognize them if they were walking down the street, are nitrification, denitrification, and ammonification. If I were to ask you which ones have to deal with a nitrogen and one of those was in the list, you should be able to say, oh, those were relating to nitrogen cycle. That's really all you need to know about it. Easy. They're all involved in that process, in the cycle in general. We have in here, we talked about potable water in here. It's not going to be on the test, but FYI, potable water is pathogen free. Not necessarily clear, might taste like junk, but it is pathogen free. That's what potable means, in case you're like out camping or something, so you know. Because that comes up. Freshwater treatment and wastewater treatment. Sedimentation with filtration, large scale, like large particle filtration. We have sludge in the wastewater treatment that we have to break down with more bacteria, and it helps us get even more and more water out of it, so we're not losing too much. Then there's more filtration. There's aeration, because if we expose some of these microbes to air, they'll just die because they can't handle it. They're not made for that. So aeration, then we put like chlorine gas through it to kill them even more. And then we can either, if you're making fresh water for drinking water, it goes to your tap, or if you're making wastewater into clean water, putting it back in the environment, that's a different thing, right? Because you can have more bacteria in that wastewater than you would in the drinking water, but you still don't want money. You want to avoid it as much as possible. You don't want to contaminate the environment. So what we do is we filter our product through a little filter paper, take that little paper after we filtered it through, put it onto an agar plate and let it grow. The bacteria will have gotten caught on it. Those, we can count those colonies, let us know how many bacteria are in there. We can even figure out which ones came from poop in general. Poopy bacteria, even if they're not the bad guy bacteria, if they are there, the bad guys could be. So that's the idea with that. So the ones that the poopy bacteria that we look for to tell us whether the pathogens could potentially be there, those guys are called coliforms, the poopy bacteria that tell us the potential of bad guys. Yeah. Food, I don't think I need to tell you guys this, right? Refrigeration is not going to kill anything, but it will slow growth. Heat can kill if it's done correctly. Here's some food from them. Now, that's chapter 25. If you guys are looking beyond that, you're way too no. If you are looking, like I went over metabolism here, I'm just like, here's these glycolysis that make the pyruvate. You're like, yeah, but they're glucose 6-phosphate boil. You don't need to know that. I didn't say it to you. You don't need to know it. So, I never even said it in the lecture, really. So, I mean, don't waste your time. If I didn't tell you guys to know it, you don't need to. Don't stress out about things you don't need to be stressing out about. Now, I'll say this. This to me, when I say these things, of course I say these things and they make sense to me. That's all I need to hear to like be like, yes, this is how the thing works. If you're listening to this stuff and it is confusing, then I would say go back and look at the more detail to see if you can understand it better. Because when you guys are filling out the answers for this study guide, I don't want you to just answer it and be like, did it, got the answers, we done. If you don't understand the answers, you will do very badly. These are not the questions on the test, right? So if you just like read this and you're like, okay, let's not start with that one. Sorry, that's not a good example. Let's go to the more complicated stuff, shall we? All right, so they're on question for microbial metabolism. We have here like define metabolism. So if you like read that and immediately in your mind, you're like, yeah, that's all the chemical reactions in the cell. Good, you're done. You don't need to study it anymore. Don't waste your time on that. Right? You knew it. Or you put it on a flashcard and waste your time looking at that. Don't. You know that one. Don't waste your time on things you know. I think that's like the biggest mistake that people when they're studying that don't know how to study, were never taught how to study, do, is wasting their time on things they don't need to know. And, you know, the teachers often assume that you do know what you need to know. Like you can parse it out and figure it out. Because we were taught how to do that. You guys weren't. So if you know a thing, and if you can look at the question and you know the answer, put that flashcard down and you're done with that flashcard. You don't need to keep looking at it. You already saw it. You're going to see the word on the test. You got multiple choice. I'm not making you write an essay about it. You'll see it. You'll remember, right? Especially if you're looking over this like the day before. Hopefully you're not waiting until the night before for all of it. But if you're just like reviewing it, then you definitely know that like you got it if you see it and you recognize it and you can get the answer right away. Yeah. However, if I were to ask like a question on there, so on here it says, oh God, I'm trying to find a good example. Yeah, so it has compare and contrast competitive and non-competitive inhibition. My question isn't going to be, you know, select the answer that is, you know, straight up define competitive or something, you know, like what's the definition of competitive inhibition, which is basically what this is asking, right? What it's going to be is, you know, pick the thing that's most closely resembles like a competitive inhibition. And it's not going to be words and words and words. It's going to be like, I have like a single word, maybe like half a sentence. I made these a lot easier than the last one. as far as less words, right? So I'm hoping that you guys won't get tripped up by words again. I know that you guys knew the information from the critical thinking, right? I'm pretty sure that most of you guys would have recognized it. But if you guys, I don't know, has anybody watched the little video I made about the critical thinking question? I recommend it. This doesn't just apply to critical thinking. It's basically going to tell you guys that, hey, number one advice, slow the frick down. It's not a race to answer that question, guys. It's not. You see the answer and you're just like, okay, this person had diarrhea. Oh yeah, that says salmonella, that's diarrhea. Okay, next one. Nope. Go back, read the question. If you haven't read every word in that question at some point, and every single choice that you have, then you're not answering it correctly, probably. You're gonna be reading the whole thing, even the short one. Yes, take the time. How many of you guys were done with your exam before the class was technically over? How many of you guys had left before the class was technically over? All of you did, by the way. All of you. Now, you feel good about that right now? If I sat you back down with those questions that we had on that last test, and I told you that you guys had, not you guys, but like other students, and I'm assuming there were other questions like this for you guys. This is a very, very popular problem. We're selecting salmonella, and that was not the answer. Why? Not because I didn't tell you the information, because I definitely talk about endospores like up and down. I talk about them all the time. So why didn't anybody see endospore and think bacillus? Why did we see, or did we see endospore and not think salmonella? And I can't really answer that for you guys, because I talk about like endospores and endospores. What bacteria did they go with? Bacillus and Clostridium. They will be the only ones that make endospore. Endospores are only made by Bacillus and Clostridium. So it's stuff like that. Whenever I see people get that wrong, and I'm wondering what happened when a lot of people did get it wrong. Because to me, that question was like endospores. Oh yeah, that's Bacillus. That's what I saw. So that's how I know it was the words. It had to be. There's too many of them. People read into like 5 words and made the choice based on that. I don't want you guys to get caught in that again, because there are going to be critical thinking questions on this exam. Don't get drowned in them. Read them. If you need to write them out in parts, you can figure out what's going on with the question. Do it. Take the time. But anyways, yes, there'll be 3 of them on this test. There'll be more on the next one. There'll be quite a lot on the last one. Probably close to as many as the first one. I went back through that first test and I realized that there were not as many critical thinking questions on there as I thought there was. I was kind of appalled. Not gonna lie. Most of them actually weren't critical thinking questions. I thought they were. I must have done something wrong, but they were not. So yeah, that was bad. And yeah, some of it was too complicated, so I'm rewriting it and going back to how I always write my tests. Taking time to go over the study guide with you guys, doing the review with you guys, trying to figure out what went wrong with you guys, making a little video to help you guys next time if you get the same question. Number one, like I said, I feel like the number one best thing you can do, especially on a critical thinking question, slow down. Actually read it. If you're just reading through it and you're not really quite understanding it, slow down. That means you need to slow down. That's the slow down, not that you don't understand. That's that you need to slow down. If you study the topic, you can know it. Right? Push yourself. You study as much as you need to. Then you won't have that problem if you slow down. I know what anxiety does to a person, believe me. So I feel like that's really what it is. People just get used to rushing through it. And it's not a race. If somebody else gets up and turns in their test, it's 50-50. So when the people get up early, either 50% of them did real shit on the test, or some of them actually did really good. Right? If you're sitting there thinking to yourself, well, I don't know that I'm necessarily going to do really good. This is like really difficult stuff, but I'm definitely not going to do shit. Then that's fine. Then don't be the first one. You don't have to worry about either way. So take your time. That's the point in all this. Take your time. Do take deep breaths. That actually does work, scientifically proven. I do it when I have anxiety. And like I will say in the video, like my therapist recommends it. That's why I do it, because it actually works. So take a deep breath if you need to. You don't need to be loud about it if you don't want to. Just do it, to your little own self. Calm yourself down. That's why I try to have the lights low. That's why I try to have like the jellyfish and stuff. I don't want you guys to freak out. I think that's the biggest thing, the biggest barrier that you guys have, freaking out. So, prove me right. Show me that you're not gonna freak out and you're all gonna get hundreds, right? Sure. Yes. Okay.